surely more of you have heard about epigenetics. And so this is actually the New Science of Epigenetics made the cover of the Time magazine, and this was in 2010. And it says, if you can read from there, why your DNA isn't your destiny. It reveals how the choices you make can change your genes and those of your kids. And so if you Google, or if you use PubMed actually, with the keywords epigenome and epigenetics, you see that 70% of the publications actually arose within the last 3 years. So there is what we call an epigenetic revolution, because a lot of people are doing epigenetic studies and are publishing about epigenetics. And Jim Watson, which you probably know as being a Nobel Prize winner for the double helix structure of DNA, actually said this summer that epigenetics is used for a lot of crap, and it's used by people who don't like genes. And he has a point there, because this explosion of epigenetics, we lost a little bit of sense of what epigenetics really is. And likewise, there is a very famous epigeneticist that you've heard from the Institute Curie that says exactly, agrees in certain extent to what Jim Watson was saying, that epigenetics is indeed a very exciting area of research. But there is a gap between the fact and the fantasy. And I think that this is the importance of starting this talk by defining what we really mean by epigenetics. And I see very young audience here. And how many of you have actually seen the Spider-Man movie? Not any Spider-Man movie, The Amazing Spider-Man. This was released in March 2012. Come on, how many of you have seen this film? More people than they actually knew what epigenetics is. So I hope that at the very end of this talk we changed this perception, okay? And so the reason why I'm saying that this is the fantasy bit is that actually Hollywood has picked up on epigenetics. And I'll urge you to actually have a look at this film and see if you can find a very subtle reference to epigenetics and epigenetic reprogramming that gives the hint that the Spider-Man was actually created because of an epigenetic reprogramming event. And so this is just a part of the fantasy about what epigenetics is. So the overview of the talk is the following. What do we really mean by epigenetics? Why is this important? And then I'm going to give you a case study from our own work of epigenetics in the context of programming of adult disease, and then end up by addressing the Jim Watson quote and also Edith Heard's about is epigenetics indeed a new field but that has been overhyped. So, when we talk to the public about epigenetics, many of us give 3 examples. So this is a poetic definition of epigenetics. What makes us different even when we are equal? And the 3 examples are the following. This, here, anyone knows what carbon copy is? It's a very famous cat. Any ideas? So it was the first cloned cat in 2001 in the US. And this is the genetic donor rainbow. And rainbow actually looks different from a carbon copy, or carbon copy looks different from rainbow. And nonetheless, this is cloning, so the genetics is the same. Nonetheless, you have a very different outcome. The other very striking example, probably one of the most beautiful we have, is these are littermates. So they are identical in terms of the genetic composition. Nonetheless, one is brown, the other is yellow, and this one is very, very fat. So it's a obese mouse. Again, epigenetics explains this because the genetics, the genes are the same. And then we have the most famous pair of twins in the world. How many of you know who these are? Come on. Very disappointing. So these are the twins from the Harry Potter movies. And it's just highlighting, in spite of them being looking the same. I'm not saying that this is particular for this pair of twins, but you can have a twin that has type 2 diabetes and the other doesn't. Or you can have one with psychological problems and the other doesn't. And so again, in terms of the gene constitution, is exactly the same. So what is this about epigenetics that explains all of this? And so one thing to remember, epigenetics, and this is probably one of the definitions that I like the most. So epi comes from the Greek that says above, so on top of genetics, on something on top of genes. And you can't even imagine the amount of discussions and papers published about what really epigenetics is, and people tend not to agree in many things. This is one operational definition. So if you can read here, epigenetics is the heritable changes in gene expression that occur without changes in DNA sequence. Okay, so a very important concept here is heritable changes, but they do not occur with the changes in the DNA sequence. In other words, inherited changes in phenotype without changes in genotype. And that's why people are so excited about the epigenetics. There are people that actually call it a second genome. So there's a lot of nonsense being said about epigenetics. But at the core of epigenetics is this. So you have the DNA, which calls to form chromosomes, and you have a nucleosome particle, which is the DNA that you can see here that is wrapped around an histone octamer. These are the histones. And so why is this important? This is important because the DNA itself can be modified. So there can be chemicals, things like a methyl group that can be put into the DNA, and we call that DNA methylation. That controls genome function. But also, the thing that really gave a very impetus to this field was the discovery that these histones, for many, many years we thought that these histones were just here as a structural protein, so that would enable the DNA to wrap around these histones. But the key finding was that you can see there's some tails here, some histone, we call this histone tails, coming out of this nucleosome. And these tails can also be chemically modified by methylation, by acetylation, and that also is going to control genome function. And so the non-epigenetic modifications, or epigenetic marks as we call it, are the following. The main ones are DNA methylation, so the addition of the methyl group, and it has been found some others related to the DNA methylation pattern. So these histone modifications, non-coding RNA. So these are the three main players of the epigenetic information. You also chromatin remodelers and higher order chromatin structure. We normally allude to those as well. So how does this actually work? It works the following way. So these are, as I said, chemicals that are added either to the DNA or to the histone tails there. And it needs to have a machinery to do that. And we normally call them the epigenetic machinery in three types of machines: the writers, the erasers, and the readers. So the writers, as you can see there, is an enzyme that actually writes the modification into, in this particular case, into the tail of the histone. And so you have enzymes that do the methylation, that do the acetylation, et cetera, et cetera. And then we have also these amazing enzymes that take out that information, those chemicals. So it takes out the methylation of the histone tails, for instance. And then you have these amazing proteins that we call the readers. And so the readers, they specifically recognize this modification. So for example, there are proteins that identify the DNA that has been methylated, and those are called the methyl binding proteins. So these are really the machinery that imposes the epigenetic modification into the genome. So very important concept here. These are enzymes, and enzymes are coded where? In the genome. So when are people talking about epigenetics and the epigenome as being something completely uncoupled of the genome is total crap. So we have to see the epigenome in the context of the genome. But what I'm trying to say here is that this is a very interesting way of controlling genome function. And we're just going to see a little bit more in detail, very basic, about what is the importance of this. So again, just to recap, you have a chromosome, you have the chromatin with the nucleosome particles, DNA wrapped around, and you have the histone tails, here, and this is the DNA itself. So as I said to you, the DNA can be modified chemically, and this is the case of DNA methylation. So it's the adding of a methyl group to the cytosine base here, and so this is called DNA methylation. This is the writer of that information, so it's an enzyme called DNA methyltransferase that puts that methylated group, the methyl group, in. And you have then histone modifications, and there's a long variety of those, so you can methylate as well, so it's quite different having DNA methylation from histone methylation, but it's the same addition of the same methyl group. You have deacetylation, the acetyl group, you have phosphorylation, etc., etc. So why is this important in terms of gene function? It's important because you can regulate genes this way. So they act more or less like a switch of on and off of genes in certain contexts. So here, for a gene to be switched on, and this is just a general picture, this is much more complicated than this, believe me. How do you have the gene switched off? So you have to have the chromatin needs to be open, so a big space between the nucleosomes so that transcription factors can bind. And this is normally associated with acetylation of histones. So these histones need to be acetylated, and the DNA can't be methylated, so you can't have methylation there as a general rule. So you have, therefore, transcription possible. When you switch off genes, you compact the chromatin, and you do that by methylating the cytosine, so DNA methylation here in red, and you need to take the acetylation from the histones. So the histones that were acetylated here need to be deacetylated, and that compacts the chromatin. So as you can see, this gives you a framework, conceptual framework, how these epigenetic marks can be so important to regulate gene expression. But the question is how often this actually occurs and how important it is. So why is epigenetics important? Because what you can see here is that we have one genome only, but we have multiple epigenomes. So you can see epigenetics as an organization of the information coded in the DNA. So one genome, multiple epigenomes. And you probably have asked this question yourself, which is, everything comes out from a single cell, which is the zygote, and you have the stem cells. And all the cells have exactly the same amount of genes. But nonetheless, you go from a stem cell to a committed cell, then to different cell types. And this is tightly controlled by these epigenetic modifications. So the differentiated process. And how is that possible? Because these epigenetics creates what we call a memory of cell identity. And so this is what the theory says, and we're going to analyze this further and probe this further and further. But how can you create a memory of cell identity using epigenetic marks? So they need to be passed on from one cell to the other. How do you do that? We know quite a lot of how it works for DNA methylation. And I'm just going to run through this with you and see if you understand. Please don't interrupt me if you don't. So you have here a strand of DNA that is methylated. So methylation is, as I said, is the addition of the methyl group to the cytosine base. And so you have it in one strand and the other strand. So these are methylated cytosines. When you have the DNA replication, of course, you'll have one strand that has the methylation and the other that doesn't. And so we call this amimethylation. So in the strand A, strand B. And then comes a very clever enzyme, which is a writer of the information that maintains the DNA methylation. So it recognizes the amimethylation here, and it just simply adds to the corresponding cytosine the methylation there. So you end up by having exactly the same pattern as you started with. And so this is how you maintain the heritability of these epigenetic marks for DNA methylation. So the heritability of these epigenetics during the cell cycle provides the means of stably maintaining a cell lineage. Now, this is for DNA methylation. But what about these histones? Nobody knows. And this is why many people say you shouldn't really include histone modification as an epigenetic mechanism, because there is no proof that they are heritable and they can do this sort of thing. There is no we don't have a mechanism by which a particular histone modification can be the same in the daughter cell and so on and so on. So bear that in mind. Although most of us say histone modifications is an epigenetic modification, that is true, but is it truly epigenetic in the sense that you have to have the cellular memory? So that's quite questionable. And this is where really people get upset in the field about all these definitions. But can we actually understand this in a very easy way? Yes, so assume that this is a liver cell. So this liver cell is differentiated and it has a fixed, what we call epigenotype. So certain genes, acetylation of histones, are normally correlated with expression, as I said, methylation of DNA with gene repression. So in order for a liver cell to remember it's a liver cell, this pattern needs to be faithfully maintained. So that's the idea of epigenetics as a cellular memory mechanism. But then the question is, what happens if you lose some of these marks? So you go from a completely differentiated cell, you lose some of these marks, some of them you lose the acetylation, other you lose the methylation. This leads to a differentiated cell. And there are many diseases, including cancer is probably the most notable of them all, that have epigenetic disruption of those marks in the cancer as compared to the normal tissue. And there's a series of other syndromes that have an epigenetic basis. Now, the second part of the second talk, I'll talk about one of those, which is an overgrowth syndrome called the Beckwith-Whitman syndrome. Now, what really has made this area go into another level, into the stratosphere, was the influence of the environment, this idea. And so this is a question mark because we know quite a lot of this in many model systems, but not really much of this is known in humans. So the question, what is the question? The question is, do these epigenetic mechanisms provide the link between the environment and the phenotypic changes during the whole of the lifetime? So in other words, you have the environmental factors that can then influence these epigenetic marking systems by unknown signaling pathways. So we call this signaling to the chromatin. And so environmental factors change the epigenetic marking of genes through the main modifications, and this leads to a change in phenotype. And you have different phenotypes here. We'll talk about the disease susceptibility as an example. But so there are two questions that remain. One of them that might seem pathetic to you is that you see weather here. I mean, come on, how can the weather modify your epigenome and your epigenetic mechanisms, and then you have a completely phenotype? But this exists very strongly in plants, and it's probably one of the best examples I can give you of the action of the environment through epigenetic mechanisms. So in particular contexts, this is actually true. But the hype about epigenetics, and people talking a lot of crap that Jim Monson was mentioning, has to do a lot with environmental factors. That the environment can change your epigenome, the epigenome will do dreadful things to your phenotype, and you'll pass this on to your children and grandchildren, and so therefore it's quite a dangerous assumption to have. The other problem I have with this is that we don't know if this is causal or an association. So of course environmental factors will probably modify the epigenetic machinery, but to what extent? And is it actually causal or is it an association? Is it something that affects the genome first and the genome controls the epigenome? And so I'm going to give you a case study based on our work, which is done in rodents, that addresses the role of these environmental factors and disease susceptibility. Okay? And I'm going to start by showing you another cover of the Time magazine and ask the same question: how many of you in the audience have heard of the environmental origins of disease? Come on, hands up. 2, 3. Okay, so it looks like you're learning quite a lot. Time magazine published this. It's again a new science, is the new science of fetal origins. And what does this actually mean? So it started many, many years ago with a very simple observation that babies that were born with low birth weight, they are at increased risk of developing diseases later on in life. And at that time, this was called fetal origins of disease. Now, these days, people have realized that you don't really need to have a birth weight difference. So this does not necessarily apply to a condition whereby you have an insult and you're born small. So you can be born normal weight and still have an increased susceptibility later on in life that is caused in utero. So this idea that the etiology of many diseases may, to a certain extent, lay in early life events that can affect later biology. So this is the crux of developmental origins of disease. And this is represented very well here, how the first 9 months shape the rest of your life. So the time that you spend in your mother's womb will be important. So this is what the theory says, and it has been it has been confirmed in human epidemiological studies, and there are many animal studies that indeed agree with this new science. And I'm going to tell you about an experiment that we did to address the role of epigenetics in this context. And so we're interested in maternal diet and offspring health. And this is actually done in close collaboration with my colleague Sue Ozan in Cambridge. And so the conceptual framework is very simple. So you have a suboptimal diet, by whatever means. This will mean that the expression of genes will change in the fetus. And then there will be a cellular memory of the suboptimal early nutrition caused by the adaptations that you do in utero. And this is the question we are addressing, is does epigenetics, as I told you, epigenetics is a very important cellular memory vehicle, and so therefore, does epigenetics play a role in this particular step? And then you have the aging process. This translates into effects on tissue function later on, and ended up by having an increased risk of the metabolic disease. And so this is my colleague Sue Ozan that we've done quite a lot of work on. And this is the equivalent to the Hola magazine or Hello magazine in Cambridge, which gives the news and views from across the university. So if you thought that we're all geeks in University of Cambridge, look at this, research horizons, that people can actually, you may recognize this dashing young man there, which is me. And in this issue was actually, strangely enough, dedicated to reproductive biology. Anyway. So what is the model that we're using, that Sue uses quite a lot? So this is a maternal protein restriction model that gives rise to type 2 diabetes. And I'm just going to go slowly about what this model is about. So this is done in rats. And what we do, or what Sue does in our lab, she has pregnant females that she gives either a low protein diet, and she has a control cohort with a normal standard diet. And so the low protein that you'll see in some of the slides will be shown here as LP, and the control is C. So we'll see LP and C several times repeated during the talk. So as a consequence of giving a maternal diet, which is low in protein, the offspring is going to be born with a low birth weight, okay? And of course, the controls will be normal birth weight. And this is done, the manipulation of the maternal diet is done during gestation and lactation, so which means that after weaning, both groups go to standard diet, standard control diet. But the effect is the uterine environment and the early postnatal of the low protein. So what happens to these here is that these low protein exposed rats, they develop type 2 diabetes by 17 months of age, so very similar to human type 2 diabetes, whilst the control never do. They have a normal glucose tolerance. So this study that I'm going to describe to you is done at 2 ages. One is the young adult, 3 months. The other one is at old age, 15 months, prior to getting the type 2 diabetes. And this is important. So we'll talk about 3 months, 15 months, LP versus controls. And so the story goes like this. When Sue first approached us, she said, Miguel, I have this cool model. Can we test that we have epigenetic mechanisms at play that is responsible for these individuals to get to type 2 diabetes and to get to increased risk, because this is really increased risk. And so what we said to her was, okay, what you need to do is to bring us a gene that is an important gene for diabetes, preferably, that actually changes, and it changes the same way during time. So in other words, a gene, for instance, that is downregulated by maternal diet, as shown here, and it maintains to be downregulated through time, which you see here. This is 3 months, this is 15 months, this is a qPCR, so you can see. So this is a gene called HNF4-alpha. And all the analysis I'm going to tell you about is on the endocrine pancreas, on the islets of the offspring that has been exposed either to the low protein or the control. So in the low protein, you can see that they make much less of the RNA than the controls of this particular gene. So you can see the difference here from controls to LP, but also during aging, there is downregulation here. It's still maintained. So what we're really interested in is in this downfall of, so from 1 to about 0.5 on the expression of this gene that is maintained at 15 months. So there is a downregulation of this HNF4-alpha with maternal low protein diet and also with aging. Low protein diet here, and then aging, you can see drops. And so what is this HNF4-alpha? It just turns out to be a very important transcription factor. So this is part of a very important transcription network, and it controls the activity of many important genes, such as the insulin, glucose transporter, etc., etc. So it's actually quite a key gene in terms of pancreatic function and differentiation. And the question we were asking is therefore, do epigenetic mechanisms, are they responsible for this memory mechanism here that goes throughout time, and is it epigenetic mechanisms that play in this downregulation of this HNF4-alpha gene? Why do we do this? Is because HNF4-alpha is such a critical gene. So mutations of an HNF4-alpha have been described in humans with type 2 diabetes, and actually mutations, I should say, this HNF4-alpha, levels of HNF4-alpha have been shown to be downregulated in type 2 human diabetic patients. And mutations of HNF4-alpha actually give rise to a very young form of diabetes in young people. So what we started by doing was, as you always do in this kind of experiment, let's look at the gene, and let's look at where these epigenetic marks are in the gene. And so the HNF4-alpha has two promoters, so P1 and P2, and this is an enhancer region. So in enhancer, what it does, it enhances the transcription of promoters. And it was well established that this enhancer acts on P1, but we also shown in this study that it also acts on P2. So two promoters, one enhancer. So this is all done in islets, and if you see here, so this is expression by RNA. Only the P2, so this one, expresses HNF4-alpha. The P1 here is completely silenced. So in islets, there's only one promoter that is active, which is P2. So we decided to look at epigenetic marks. How do the epigenetic marks look at this region? So if you see here P2, so these white lollipops mean there is no methylation. The black lollipops means there is DNA methylation, so cytosine methylation. And you can see this overall, this is rather unmethylated. So unmethylation means that the gene can be transcribed. But now if you look at the P1, it's just the reverse. So there's a lot of methylation, and there is no expression. So this correlates beautifully. P2 is unmethylated and is expressed. P1 is not expressed in this almost fully methylated. So beautiful correlation with the epigenetic mark, which is DNA methylation. And we looked at histones. So we have ways of looking at the enrichment of histones at these regions. So one is P2 here and P1 here. And we also looked at the enhancer. And I'm not going to bore you with the details of which marks these are. So these are these three, the green ones and the yellow are active marks, and the other ones are repressive marks. So you can see that the P2, there's an enrichment for the active marks, which is totally consistent with P2 being the expressed one. The enhancer is also open chromatin. In the P1, you don't have much of, you actually have repressive marks here. And how do we know that this is actually a functional enrichment? Is that we have control genes. This is actin B, and this is hemoglobin. And you can see that this is the positive control for activation with histones, and this is positive control for silencing, where you only see the repressive marks at play. So a beautiful correlation between epigenetic marking systems and expression of the genes. So we're really happy and enthusiastic about this because DNA methylation, so this is only control islets. So DNA methylation was really, in particular, very well correlated in histones as well. So now the question was, let's analyze the DNA methylation in the controls versus LPs, because as you remember, the LPs downregulated the HNF4 alpha gene, and therefore we were expecting to see an increase in DNA methylation, which would then be nicely linked with the downregulation of this gene. And so that's exactly what we're trying to explain here is this downfall here between the controls and the LPs there. And when we did it at 3 months and 15 months, you do see a slight increase in DNA methylation here. But this is an increase of methylation that is too small. You can't possibly say that this difference in methylation can be responsible for 40 to 50% downregulation of a gene. So DNA methylation was really a disappointment, especially for the postdoc that did all this work. So the P2 promoter, you see a little bit of increase, and in the enhancer regions there is no difference. Also, this doesn't explain the downfall during aging. So rather depressing times for the postdoc. But then we had made this very interesting observation about the enhancers. And so I'm just going to go through this experiment with you. So this is the control islets, and these are retinosolinoma cells. You have two types. And what struck our attention was the fact that you have, so this is the mRNA of HNF alpha for the controls, for the insulin cells, and for the brins. And so here, I've already discussed this with you, the activity of the marks, also open chromatin of the enhancer. And the P1 is not that enriched at all. But if you compare now the control islets with the insulin 1 cells, you see that there is a drop in the mRNA, and this associates quite nicely with the repressive mark that enriches the enhancer. So in other words, the P2 promoter seems to be fine, but what you're starting seeing here is a closed enhancer chromatin. And in the brain cells, everything is really, really completely compacted with repressive marks for histones. You don't see any expression. So this really told us maybe the key of this is at the level of the enhancer promoter interaction. And that's what we explored further using a very cool technique called 3C. And again, I'm not going through the details, you can discuss this later. What this allows us to do is to measure the distance between the promoter to the enhancer. So these things are not aligned. So these things, they actually loop. So the enhancers and the promoters, they come together. And we've decided to look at two tissues. One tissue, which was the liver and the islets. Now the islets, as I told you, is the P2 promoter that is very, very high in transcription. P1 is very, there is no expression of P1, basically because it's methylated. And you can see that by doing this 3C, the P2 comes very close to the enhancer. So therefore, it's no surprise that you have P2 expression. As I said, P1 is not expressed because of DNA methylation. But if you see the liver has the opposite pattern of mRNA. So here is the P2 is silenced, and the P1 is quite highly transcribed. And when you look at where the P2 is in relation to the enhancer, it's much further apart. So this might well be the explanation why P2 is completely silenced. So it has to do with the enhancer promoter, how they come close together. And so then we decided, okay, what will be cool to do is analyze this chromatin looping in the controls versus the LPs. And what we found was pretty amazing, which is, so this is the control diet, where you have the enhancer next to the P2, as I showed you in the previous slide, a lot of expression. But at 3 months of age, the low protein diet, you acquire enhancer-specific epigenetic modification. So you have recruitment of repressive histones, you lose the active ones, the two active ones. So you have this red flag, which means silencing, and this makes the P2 going further apart from the enhancer. And this is very nicely represented here, the control and the LP. So this is the strength of the interaction. You can see controls a positive band in the LPs, you barely see it. So which this means is therefore that maternal diet or early diet has an effect on the enhancer-promoter interactions. And to us, this was really exciting as a novel epigenetic mechanism that is under the action of the early diet. And so what occurs with aging is actually quite also very interesting. So this is the young adults, what happens in the low protein, that the enhancer-specific epigenetic modifications make the distance between P2 and the enhancer being more distant to each other. During adults, now not only you have the same enhancer being recruited for epigenetic modifications that are repressive, but you add a very strong repressive signal, an histone mark, which is H3K27 trimethylation. So this means histone 3 is methylated ME3 in 3 sites at lysine residue 27. That's what this nomenclature means. And so you have more of this in the enhancer, but also the P2 becomes now silenced. And so during aging, you have an epigenetic silencing through histone modifications. And in the low protein diet, this is exacerbated, this recruitment is exacerbated in the low protein diet. You have more of this repressive one, which would probably mean that we'll be even further apart. So what are the conclusions of this? So these are the following. So the maternal diet and age reduces HNF4 expression, and I think I've shown you this quite clearly. We think that is mainly, the mechanism is mainly epigenetic. What was interesting about this study is that it does give you a hint that we should be looking at regulatory regions when we do epigenetic studies. So don't just do an epigenetic study that is focused on promoters, because you also have these enhancers that are important for gene regulation. There's evidence for age-associated epigenetic silencing, which is really being driven by the polycomb-dependent histone mark. And we think that these changes may explain why you have this link between the poor diet and increased risk of diabetes, because HNF4 is such an important gene involved in diabetes. But to me, the most interesting aspect of this work is that it places the enhancer-promoter interactions as being a potentially novel mechanism by which you can regulate genes. So when you have a situation of cancer and epigenetics, you have all or nothing. But in common diseases, what you need is to have very low levels. You can reduce a gene at very low levels, and many genes reduced at very low levels, at some low level that is not black and white as the cancer is. So it's a very interesting way of modulating levels of genes by altering the promoter and enhancer interactions. And so this is something that we want to prove, that this is a general mechanism by which nutrition can alter expression of genes and be responsible for the increased risk, is by looking at all the enhancer-promoter interactions in the genome. And how do we do that? Well, this is really how we felt at this stage, because on the earth, are we going to do that? You're a bit of a bugger, you need help, and when you need help, you contact your friends. And luckily, we have a very good epigeneticist at the Weber Institute just around the corner in Cambridge that does a very interesting way of capturing all the interactions between promoter and enhancers at a genome-wide level. And I'm just not going to bother giving you the details if you're interested. But in other words, there was a Nature paper that was recently out that about nearly half of the mouse genome is organized into the enhancer-promoter units. So the genome is tightly regulated by these enhancer-promoter units, but nobody has been looking at them in a genome-wide level. And this will enable us to find these interactions, long-distance interactions between a gene that sits here and another regulatory region that sits here, both in the same chromosome, but also in different chromosomes. And so for the next, how much time do I have? 5 minutes, 10 minutes?
5 minutes.
5 minutes. So I'm just going to go through this idea is epigenetics overhyped, and very quickly? Question number one, this that made cover of Science is the fact that you have one genome, undifferentiated cell, and you have differentiated ones. You have the multiple epigenomes. I said to you that this can be explained by epigenetics, which is this is the stem cell. These are all different looking cells that derive from this one, and they all have different epigenomes. And we know that this is the case because epigenetic regulation of pancreatic development or even adipogenesis, we have identified important key players that are epigenetic in origin. So this is a writer for this mark, H3K27, which is an inhibitor of the beta-catenin that promotes or that inhibits adipogenesis. And in the pancreas development, we have identified many of these writers and erasers of epigenetic information, some of which are very important for the cell deciding if it goes that way or if it goes that way. But then it comes the people that say, oh, this is rubbish because it's all down to transcription factors. Why do you need epigenetics marks? It's all down to transcription factors. And their biggest argument is these beautiful experiments that were done with transcription factors. So you can take a fibroblast, you put MyoD in it, a transcription factor, you can get muscle cells. You can get an exocrine cell, you do a cocktail of 3 transcription factors, and you turn them into islet cells. And so even the biggest argument is when you get fibroblasts, and this was the experiment that gave a Nobel Prize to iPS cells, inducible pluripotent stem cells. Get fibroblasts, you have a cocktail of 4 transcription factors, and you get a completely embryonic stem cell, a pluripotent stem cell. So the people say, why do you spend time saying that it's all about epigenetic mechanisms when I can do all the job using transcription factors? The key is, this is not a very efficient process. You need to screen thousands of these clones to get one that goes right. And that's exactly because you need to reprogram the epigenome. So you need to wipe out these epigenetic marks in order to be able to get this to be a success. So that's the version that tells us that the epigenetics is important. And we have this epigenetic information being very dynamic during development. So there's an amazing, so this is for DNA methylation. There's an amazing erasure of the DNA methylation at 2 stages during embryonic development. It's quite dramatic and dynamic. So this is in the embryo and also in the germ cells. So you need to wipe out these previously acquired epigenetic information so to acquire totipotency. So they need to be reset or reprogrammed in early embryos and in germ cells in order to achieve developmental pluripotency. And so this is quite a key aspect of epigenetics. It's quite dynamic at different times of development, but at the same time, it needs to be very robust and very stable. And this is where all the confusion starts with people writing that, you know, this is the epigenetics, genes have a memory, but you know, whatever your grandparents have done during their life can actually affect you guys. And this is where you become to fantasize about epigenetics because of this idea that the environment can create havoc to the epigenetic marks in the environment. But there are beautiful examples how this works. So there's certain plants where in order for them to flower, they need to be exposed to cold. And these cold responses are going through epigenetic modifications. But to me, the most interesting one is actually what happens with the honeybees. And so, as you know, there's very few queen bees and a lot of worker bees. And so these are sterile. This is the only one with reproductive properties. And the difference is that only the queens, they are fed this royal jelly. So they have this super sort of food in the form of royal jelly throughout their lives from larvae, and the workers don't. And so the fact that she is going to be a queen has to do with nutrition. Now, what this study has amazed the world was by showing that this nutrition modulation goes through DNA methylation. And the key experiment that they did was at the larvae stage, they erased the DNA methylation, and larvae that were going to be sterile workers suddenly started to become queens. So that's an amazing example of how manipulating DNA methylation can have such a dramatic effect on reproductive status. This, guys, is environment. The problem is when we talk about humans, all of this goes a little bit out of control because we don't really have much of evidence. Possibly another wonderful example is given by the agouti viable yellow mice. I started by showing you that these mice are identical. So this was one of the very first slides. One was yellow and obese, the other one was brown. Actually, it's more of this color. And this has to do with the fact that there is a variable methylated region upstream of this gene here, that if it's going to be methylated, this is only expressed in the skin cells, and you get this color. But if this is not methylated, the agouti gene is going to be expressed everywhere throughout development, affects the connections of the brain that controls society behavior, and this mouse just can't stop eating, and that's why it's yellow and is obese. It has to do, all to do with this methylation here. Methylation controls the activity of this gene. If it's unmethylated, you get the obesity phenotypes. And there are very cool experiments whereby you can supplement a particular cross with methyl groups, and you totally change the phenotype, the expected phenotype. So if you have a little bit of methylation, but still left, you get these kind of mosaic colors here and here. So this is a gradient. This is fully methylated, unmethylated, and partially methylated. If you supplement with methyl groups, what you do acquire is a shift in terms of coat color. So instead of having the proportion here, you start having much more of the brown ones, which indicates that you get a shift into methylation, and therefore you get a beautiful readout, phenotypic readout in the skin by using a maternal diet. And so it's literally the last slide about this area of nutri-epigenomics, and maybe the title of this should include epigenetics. So there's clearly evidence that diet influences the epigenetic marking of genes, but we know really very little about the mechanisms. The epigenetic changes can occur through altering the availability of the substrates or altering the expression of these writers and enhancers. In particular, these are just the cofactors that we know that are very important for the epigenetic enzymes to work. So you can see that there will be a problem if you have the levels of these modified, so altering the availability of substrates. And there are very cool experiments that people have done by knocking out the particular, in this particular case, it was an eraser of methylation of histones, this particular gene here. And if you knock it out, what you get is an obesity and a very strong metabolic phenotype. So it can really address the functional consequences of this. And so we really need to stop saying that, you have an environmental influence that changes your gene, and that is epigenetics, because epigenetics has this idea of cellular memory. Don't forget about that. And we need to be careful about what we say regarding transmission of epigenetic marks and transmission of the environment. And that's a particular quote that I found in a scientific website, which is not really very helpful. Thank you very much for your attention.